Now we reach the last lecture in our session, and this is, I'd like to present Professor Layla Montaser, professor in Mufayna University. The title of her presentation is Stem Cell Therapy, Expand the Horizon of Regenerative Medicine.
Bismillahirrahmanirrahim. Thank you very much for Professor Dr. Amal for inviting me to contribute an oral presentation at her elegant conference. Bismillahirrahmanirrahim. My presentation today titled Stem Cell Therapy, Expand the Horizon of Regenerative Medicine. You can see the logo. This is the logo of our group of CERN. CERN means Stem Cell, Regenerative Medicine, Nanotechnology, and Tissue Engineering Research Group. Outline, introduction, introduction, and brief synopsis draws on some research studies on thalassemia, and then I will present some of our in vitro and in vivo regenerative medicine studies. Regenerative medicine is a new paradigm of medicine that relies on stem cells' capacity to regenerate, regrow, and fully heal damaged tissues and organs. It is a branch of translational research in tissue engineering and molecular biology, which deals with the process of replacing, engineering, or regenerating human cells, organs, or tissues to restore or establish normal function. Mesenchymal stem cells have been one of the most intensely studied candidates for cell therapy. Stem cells have the ability to develop through the process of differentiation into many different types of cells, such as liver cells, skin, brain, or lung, etc. One of the big challenges of using stem cells as a therapy is coaxing them to grow into the specific type of tissue that is required. السيل هي بتعمل engraftment for herself. The increased interest in stem cells and their therapeutic potential leading to a new era in regenerative science. Today, stem cells are used to treat a variety of degenerative conditions such as blood cancers, blood diseases as thalassemia and sickle cell anemia, diabetes, cardiovascular disease as congestive heart failure, pulmonary disease as COPD, erythema, and idiopathic pulmonary fibrosis, and some degenerative neurological diseases. Also, orthopedic diseases or injuries like arthritis, knee injuries, degenerative hips, support injuries. My interest, liver, whether acute or chronic, and also in the degenerative disease, osteoarthritis, knee osteoarthritis. A stem cell is a single cell that can replicate itself or differentiate into many cell types. Let us go to synopsis draws on some research studies on thalassemia. I will run quickly on these. This paper, Stem Cell Treatment, Holy Grail of Thalassemia Management. I get this paper only May 8, since less than 48 hours only. It is published online, not in journal. That's why this is the URL for this paper. The importance of this paper, this professor at India, he met like the Akhi with center in the United States of America at California, San Diego. But this for the professor, the Indian professor. They have recently launched the first CliniMax Prodigy system in India at their center. A center that in India, the other one at United States, California, at San Diego, which I visited several times. With the hope of being able to effectively treat thalassemic individuals and also those suffering from other hematological disorders or malignancies. The just hope, for me, my hope was since 20 years, my hope was for stem cells. I like stem cells too much. My hope was since 20 years. Thanks to God, alhamdulillah, for everything after how many years. The aim, the Indian doctor, is to reduce the waiting period for transplants and bring down the rate of mortality among affected individuals of thalassemia. With a combination of mesenchymal cells for restoration of the bone marrow microenvironment and the CliniMax Prodigy system that الاسم ده at India and at the same name at San Diego. Individuals suffering from thalassemia will be able to lead a normal life free from major side effects associated with conventional modalities of treatment. This second, I selected the most recent papers. The first one was since 2 days only, 8 May 2018 or 2018. This paper published at Blood 20 or 2017 under the title of Impairment in the Mesenchymal Bone Marrow Niche of Beta Thalassemia Patients and the Association with Prolonged Iron Exposure. Data on mesenchymal compartment in beta thalassemia patients are scarce, of course. I think maybe no need to present.
Say the most important points.
Yes, they isolate and characterize mesenchymal stem cells from beta thalassemia patients and healthy donor bone marrow samples. Beta thalassemia mesenchymal stem cells showed reduced clonogenic capacity, delay in colony formation. They demonstrated that mesenchymal stem cells are able to uptake and store iron. In conclusion, they said that they showed an impairment in the mesenchymal niche of beta thalassemia bone marrow, possibly associated with prolonged iron exposure. This is the last one I selected. Bone marrow mesenchymal stem cells in patients with beta thalassemia major. This study aimed to investigate global structural and compositional changes in bone marrow mesenchymal stem cells in beta thalassemia patients that may provide basis in understanding interactions of hematopoietic stem cells and mesenchymal stem cells in such pathological bone marrow microenvironment. The results provide information for the studies that concentrate on interactions between hematopoietic stem cells and mesenchymal stem cells in the bone marrow. A regenerative treatment model. In this model, stem cells mediate a repair via 5 primary mechanisms, providing an anti-inflammatory effect. Second, homing to damaged tissues. Third, supporting tissue remodeling over scar formation. Fourth, inhibiting apoptosis. Fifth, differentiating into bone cartilage, tendon, and ligament tissue. In children, the stem cell therapy permitted in different types of blood cancers and in some non-cancerous diseases, such as thalassemia major, juvenile rheumatoid arthritis, and osteoporosis. Let us go to our in vitro and some of our in vivo regenerative medicine studies. This our study at Menofeia, at Clinical Pathology Department, Faculty of Medicine, Menofeia University, Egypt. This our study. Our team evaluated the visibility and the efficacy of adult stem cells to be separated and differentiated into functioning hepatocyte lineage. The successful rate was 92.9%. But this human bone marrow samples, we use human bone marrow samples. We start separation and culture, expansion of mononuclear cells, then culture to mesenchymal stem cells, then to hepatocyte-like cells, to hepatocyte lineage. Sampling of induced and non-induced groups for morphological examination observed under the in vitro microscope, reverse transcriptase PCR analysis of hepatic gene expression of non-induced and induced groups of bone marrow mesenchymal stem cells. In this study, PCR was reverse transcriptase analysis. A new study, now not yet finished, real-time PCR. There is a paper I reviewed at Cell Journal. They used fetus of rat to induce liver disease, and then they treat. They open the abdomen, and they took the uterus and the liver, and but this under revision. I accept her, but unfortunately, the test reverse transcriptase PCR, and I advised the authors. My comment to the authors that this effort was better for real-time PCR, not for reverse transcriptase PCR. But my comment to the editorial to accept the paper. I did not include here because this is not yet, but they will publish. Yes. This is inverted microscope images of morphology of bone marrow cultured cells. This day one, after first staging, we can see rounded adherent cells. This under inverted microscope. All the figures under inverted microscope. We can see fusiform fibroblast cells with many, of course, rounded cells. This on the 7th day. On the 10th day, we can see increased fibroblastoid cells and confluence to about 30%. This figure on the 12th day, you can see increase the fibroblastoid cells with about 50% confluence. And of course, less rounded cells. We can see only some few rounded cells. This at 15th 15th day, increased fibro. We can see increased fibroblastoid adherent cells with the confluence more, about maybe 80 to 90% confluence. This, the last figure for this group. This at day 4. For the differentiated mesenchymal stem cells, we can see some change in the morphology in mesenchymal stem cells to round and epithelioid shaped in between the fibroblastoid cells. This, we can see this differentiated mesenchymal stem cells on the day 7. We can see morphological change في mesenchymal stem cells to, we can see polygonal hepatocyte-like cells. This, you can say, hepatocyte lineage. This flow cytometric analysis of surface protein markers of bone marrow mesenchymal stem cells. We can see this surface antigen markers. The conclusion. Yes. This, دي اللي أثبتنا بيها إن دي hepatocyte lineage. دي flow cytometry ودي PCR وده الألبومين time dependent one. At Guangzhou, China, I presented this paper. Bone marrow mesenchymal stem cell showed immunophenotypic marker positivity for CD45, CD34, and CD14. This is another study. The other one only for preparation, or this is regenerative medicine study on acute liver failure. This on the rat. Oh. Yes. Yes. The result of flow cytometric analysis of human mononuclear cells. You can see positivity for CD45, CD43, and CD14. This result of flow cytometric analysis for rat mononuclear cells. This is schematic representation of the process of tissue regeneration in liver tissue engineering. This I presented at San Diego, California, and also at Royan International Congress at Tehran, and published in international journal. This is my photo during at San Diego. This drug intraperitoneal injection. This histological results of the study groups. This is steps of preparation of platelet-rich plasma, which I prefer to call plasma rich in growth factors. The platelet-rich plasma injection, intra-articular knee from lateral approach. الرسالة دي, this master science thesis, yes, yes. I published an international journal of advance in tissue engineering and regenerative medicine. Received citation report from the journal that this article captured vast citations all over the world. In conclusion, mesenchymal stem cells represent an exciting progenitor cell source for applications of tissue engineering and regenerative medicine. The complexity of the regenerative science will require a significant period of further research. Regenerative medicine and the interventional clinical pathology and hematology will change everything. This, my group, and the rest, I don't have their figures, but two of them, Dr. Jihan and Dr. Marwa, at Saudi Arabia now. Thank you.
Thank you, Dr. Layla, very much for this innovation, really, in stem cell and mesenchymal cell science and elaboration in the regeneration of tissues. Thank you very much.